you want an extra challenge? An extra little piece? If you want to learn better from a lesson, every time I write a sentence, write it in your own words. Don't write what I write your version of what I write. Does that make sense? That will take this lesson and it will immediately make it stick in your brain twice as well. It's a really good strategy. However your teacher writes a note up there, read it, synthesize it, and listen, if you're like, ooh, I'm not sure I 100% understand that, I don't want to try putting it, then just write it as is. But if you're like, ooh, yeah, I get that, I know what we're talking about here, then take the challenge. Don't write what I write. Write it your own way. That helps cement things. That'll help it stick in your brain. All right, so we're going to talk about DNA. And the reason we're going to talk about DNA is because, A, you can't talk about genetics. You can't talk about how your DNA interacts with stuff. Let's see in a few minutes. This is a long one. Stick around. Nice to see you. Sorry, I'm going to sneak by. I'm going to do the thing where I stand up. Do you imagine having to sit for like a whole 75 minutes? Can you imagine what that would feel like? If it was just like, oh my God, there's just somebody talking and they're stuck in one spot for 75 minutes and you can't get up and move around. It'd be torture. It'd be against the Geneva Convention. All right. He can. Okay. I'm sorry. I've been told that. All right. So let's take a look. We want to talk about DNA, and first and foremost, we'll just say what we already know. Oh, why do I keep doing that? Oh, I never thought of that. But this is like. It's like when my wife is stressing about something and I tell her that she should calm down. She's always like, oh, that's good. I hadn't thought of calming down, and now I'm going to do that because the only thing missing was a man telling me that I should be calmer for me to know how to handle this situation. So all cells contain a genetic code, which is basically a blueprint for that organism. And this is stored in a molecule called DNA. And, all right, who's the keener? Who read ahead? Who knows what DNA stands for? Anyone? It's okay if you don't. I'm about to tell you. That was terrible. That wasn't even funny. Advice. What's advice got to do with it? Anyway, that one hurt my feelings, but it wasn't even good. It more hurt my feelings that you've been with me all this time and that's all the wit I've passed along. For shame. I will try harder moving forward. But it's kind of interesting. You've had this. You've probably known about DNA since you were what? Like, I don't know, 6, 7, 8. Like you hear about DNA early on, right? Okay. Well, the D in DNA stands for deoxyribo. Now, off to a good start. Makes perfect sense. Nucleic, and the A stands for acid. Deoxyribonucleic acid. So you can obviously be excused if you didn't know what DNA was, because that's a pretty esoteric term, right? So what does it mean? Well, I'm going to tell you. So the first thing we'll say is that DNA is a chain, and each link or each piece is called a nucleotide. If you're a chemistry buff, one of the things we call this is we say it's a polymer. It is a molecule that's made up of a chain of the same molecule over and over again. So DNA is a polymerized molecule. And that polymer, that piece, is called a nucleotide. And the nucleotide itself is made of 3 parts. And we're going to draw it. And the first thing to understand is that this is a sugar that is made of 5 carbons. All right? It's called deoxyribose. But the main thing to understand, and of course I was going to draw that in green because what color is it in the name there? Green. Come on. There we go. This is a 5-carbon sugar. Now, of course, how does your body get sugars? Sorry? Eating, yeah, plants. So plants make sugars, we eat them. And one of the things those sugars can be made into is this part of the chain. It is attached to what's called a phosphate group. And the phosphate group actually isn't part of the name, but it's an integral part of this. In fact, here on one end, we have what's called a phosphate group. PO4, that's a phosphate group. I'll look forward to it. Sorry about that. And then finally, we have what's called the nitrogen base, or the nitrogenous base. Now, these two components of DNA are always the same. In other words, one of your chromosomes has hundreds of millions of individual pieces of links in the chain of its DNA. Every single link has a deoxyribose sugar stuck to a phosphate group. It's this last piece here where there's variability. Now, we use a code every day to carry on information, right? We pass on information. We're doing it right now. We write words. How do we write? What do words represent? Kind of esoteric, but they represent concepts, right? Like you put a C, an A, and a T together, and you sound it out, and you say cat, and you teach a little kid this, and they know that that 3-letter word makes the sound cat, and they know that cat represents like a living thing that actually is a cat, right? And you just take for granted that this is something we do. It's actually incredibly sophisticated. Humans naturally develop linguistic speech. Writing is not natural. If you leave a group of young children together, and this is true, this has happened over and over again, they will actually spontaneously develop language. In fact, if you put a bunch of kids together in migrant communities, they will naturally form a spoken language all themselves. Do you know what we call languages like that? These have famously happened in places of colonialism, where you get, like European nations come in. So say like Haiti, the French come in and they take over a place and they, steal everything from the locals, put the locals to work. But then also this is the time of like the African slave trades, and they also bring, people from different countries there. And all of a sudden you've got this ruling class of people who all speak French. And then working for them in their plantations, you have people from different countries, different communities who speak different languages now stuck together. And their kids naturally form a language. That's exactly how the Haitian language came to be, but that's actually how hundreds of languages all over the earth came to be. Do you know what we call that type of language? Creole languages. You heard that word before? Creole isn't a specific language. Creole is a type of language. Creole languages happen when communities form where basically the adults in the community all speak different languages, and their kids will just naturally grab words from all the local languages, but it develops its own grammar, and it happens just automatically. The human brain is hardwired for spoken language, but the human brain isn't hardwired for written language. Literacy doesn't come naturally. You have to be taught it, and kids will not develop it if they're not taught it. It will be developed over thousands of years, but it's actually a really sophisticated thing, right? Like each symbol represents a sound, and if you put the certain symbols together, that makes a word, and each word represents an idea. Now, when we use our code, our written language, how many letters do we have to work with? 26. What about computer? Computer's working code. How many letters do they have to work with? Does anyone know? Anyone take coding? 2. Yeah, binary, 0 or 1. And everything your computer tells you, it does so using just 2 letters, either on or off. It's traditionally been done with magnetized. This section of the disk is magnetized, that section isn't, so as it gets read across 0, 0, 1, 1, 0, whatever. And that's all the information they have to make complicated things, pictures, videos, texts, programs. So, how many letters in DNA? How many different letters are there where we have the bases? There's 4 possible bases. There's only 4 letters in the DNA alphabet. All right? And they are, one is called adenine. That's one of the bases we can have here. The other is called guanine. The third is called thymine. and the fourth is called cytosine. These are ring structures. These two are similar to one another. We call these the purines, and these are a double ring structure. And we call these the pyrimidines, and they're a single ring structure. Eventually, you will memorize this, I promise. In the short form, or in the short term, you don't have to memorize that they're purines and pyrimidines, but it's noteworthy that there are 4 different molecules that can be shoved on here that are all similar to one another, but still different from one another. Does that make sense? They're similar enough to each other that they can be stuck on here and stay there, different enough from one another that a molecule that's designed to come up and move across them can react to each one differently, which means that they can cause the machines of the cell to react differently to adenine than guanine than thymine than cytosine. And that's the foundation of the genetic code. The idea that as we put DNA together, we can get that. I'm going to draw these. Am I going to draw these, or will I just give you a handout tomorrow? Shh. I'll just give you a handout. Since Kaylin told me, and then I said her name wrong. So a strand of DNA is just a chain of nucleotides. Now, that's a little bit like saying a book is just a big, long string of letters, one after the other. It's true, but it hides an important piece of information, right? If you open up your favorite book, I've got a stack of books I've read in the last few weeks sitting over here. If I picked up the top one there and started looking through it, it's just letters, one after the other, right? And some are spaces, but if you think of that as another character, right? How does that chain of letters become information in a book? How does that chain of letters become information in a book? You learn how to read it, and you look for combinations of letters, right? And those different combinations of letters mean different things. So a strand of DNA is just a chain of nucleotides, and then the bases in groups of 3 called codons form a code. With the codons, matching with different building blocks of proteins in the cell. And let's pause, because that's a big sentence. So first of all, write it, and then let's talk about what it means. Or maybe we'll talk about what it means, and then you write it in your own words. What the hell does that mean? The bases in groups of 3 called codons form a code, with the codons matching with different building blocks of proteins in the cell. What this is telling you is that DNA is used to tell the cell how to build proteins. That's the information that's in your DNA. The instructions on how to build proteins. So let's take an example. A, what's that stand for again? Which base pair? Or which base? Which of the possible bases? Adenine. And then let's say we had a G, and then a G, then a C, and a G, and a T, and an A, and a G, and a C. And this could keep going. But each set of 3 gets read by the cell, and this codes for the building block of a protein, which will get stuck to the next. I could draw these a little bigger and they'd go. That says, hey, that's for this building block. But then this code here, that actually pairs for this building block here. Does that make sense? And then AGC, actually, I'm going to go back and say it's AGG again and show that, that's that same first building block again. Does that make sense? And these just get stuck together. And so what the cell does is it reads the DNA and it uses it to build a protein. That's what your DNA does. If you take grade 12 biology, or if you took the grade 11 college biology, we actually look at the code. We actually learn how to read the code of DNA and how the cell says, okay, this is the code of that DNA, this is what that protein will be. For now, we're just going to learn that that happens. I can move that up. Now, as I'm sure you can imagine, this process is really complicated, right? The process of having a machine in the cell that goes along the DNA, reads it, and uses it to build a protein is a complex machine. But I hope you see that, at its most basic, it's fairly straightforward. Basically, the DNA code has an alphabet with 4 letters. And every word is how many letters long? 3. We call each word a codon. How many possible codons are there then? Anyone taken data management yet? Probably not. How many codons are there? Right, if we go all the way from AAA to AAC, whatever, all the way up to TTT, how many combinations are there? Well, when you pick your first codon, how many choices do you have? Could be A, G, C, or T. How many choices is that? 4. And then for each of those, we could branch off, because how many ways can you pick the second one? 4. And then how many ways can you branch the third one? 4. So the other thing we could say is that's 4 choices made how many times? 3. What's 4 to the power of 3? 4 times 4 is 16. And 16 times 4 is 64. Imagine if your language only had 64 possible words. That means there's only 64 possible ways that your cell can say what the next building block is, which means your proteins can't have more than 64 different building blocks. It actually has much less than that. Does anyone know what the building blocks of a protein are? Don't worry if you don't. We'll do a whole unit where we talk about it. Sorry? Amino acids. Very good. Does anyone know how many different amino acids there are? There's only 20. So actually, the code is overkill. It gives us duplicates. Sometimes 2 different words code for the same amino acid. But that's it. There's only 64 possible ingredients in your proteins. That may not sound like much, but keep in mind there's only 12 notes in music, and we've made a lot of different sounding music, right? So you can do a lot with 20 codes. But that's it. For example, just to give a sense, human chromosome number 1, your chromosome number 1, is about 250 million, is about 249 million base pairs long. So a little bit less than 100 million codons. How's that compare? Like just, okay, what have I got over there? Stop book. 2 Solitudes, Pierre Burton. How many letters would be in a typical book? I don't know. When you write a long essay, what's a long essay for you guys? Like in English, maybe a 2,000 word essay, would that be a long essay? Sorry? Yeah, 2,000 is pretty long, right? That's not nothing. That's a few pages, yeah? How many letters would that be? Well, how long is the average word? Maybe 5 letters, more or less? That'd be like 10,000 letters, right? Compared to 249 million. That book would probably be about 20 or 30,000 letters. Does that make sense? Compared to 249 million. And 249 million base pairs isn't in your whole DNA. That's just in one of your chromosomes. How many chromosomes do you have? 46, yeah. Now, that's actually the longest chromosome, but still, to give you a sense, a lot of information. Yeah?
Yeah, I'm just going to do what Mr. O'Donnell. So, the bunch of million base pairs, like how we had AA.
Yep, that's right. So chromosome number 1 would look like A, C, C, G, T, C, G, A, C, T, G, C, A, dot, 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 T, C, A, G. And all together, there'd be about 249 million of these things.
Like pairs of them?
We'll talk. Oh, we haven't talked about pairs yet. Sorry, we'll talk about pairs. Sorry, that's the next note. Yeah, it just says nucleotides in my thing. Sorry, don't worry about the word pairs. We'll get there in a minute.
So it's that many letters.
That's right. Yeah, compare this to a novel, which is, depending on the novel, which might be 200,000 letters long. Just substantially shorter. A long novel might be 500,000. Still nothing compared to just, 500 times that much in just one of your chromosomes. So we're talking about a tremendous amount of information. And that is a product of evolution. Did the earliest cells have that much genetic information? Absolutely not, right? That's happened over time. Okay, now, what's DNA look like? That's good. What's it look like? Ha! I like that. A lot of hand motions today. Like a spirally ladder. I like that. It is like a spirally ladder. In fact, we say shape-wise, I'm going to make a subheading here. Shape-wise, the DNA molecule has a, and you may have heard this before. Tell me if you have. A double what? Very good. A double helix shape. But you're right. We tend to draw it, and so we tend to, ready for some amazing art? We tend to, oh gosh. Oh, this is going terribly.
Oh, looks great.
I don't need your pity compliment. Yes, I do. Tell me it looks great.
I've never, it's better than Picasso. That's the name, right? Picasso?
Okay, there we go. And it's got this kind of structure that looks like this. Okay. But here's the thing. Double? What do you mean double helix?
Aren't they like, if you look at like a colored diagram, aren't they like two colors?
If we look at this section of the DNA right over here, that would look like phosphate, sugar, all right, and then A. And then that connects to the next phosphate, the next sugar, and then G. And then the next phosphate, the next sugar, and the sugar connects to T. And then the next phosphate, and the next sugar, and that connects to C. And that would carry on in both directions. Does that make sense? Okay. But this says that it isn't a single helix. It says that it is a double helix. So what is this? Well, this must also be a phosphate connected to a sugar, and that one's going to connect to a G, and we'll talk about why I picked G in a minute. And that's a phosphate connected to a sugar, and that's going to be A, and we'll talk about why I picked A in a minute. And that's a phosphate connected to a sugar, and that's going to be, oh, and we'll talk about how I picked wrong in both cases. T, C, we'll put an A here, and a phosphate connected to a sugar, and we're going to put a G here. Okay. And we'll talk about why we did that. But the key thing to note here is that DNA is always double-stranded. How many of you are in chem right now with me? Okay, if you're not, you can just la 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 la. Okay, These molecules here are highly, highly, highly polar. And on one side, they have an extremely polar region. And what you get is a massively strong hydrogen bond between them. All right? Don't worry about whatever. But the point being, these base pairs are designed to form really strong, really solid bonds with each other. And they're not just designed that way, but they're designed such that they pair up in predictable ways. So they always pair up base to base, and such that A always pairs with T, and G always pairs with C. So if there's a T on one strand, what's going to be across it on the other strand? Very good. If there's a C on one strand, what's going to be across it on the other? Very good. Okay, I have a very clever way of remembering these. Always together. Huh? Yeah. And always together. I don't know. But that's how I remember that A and T are always together, is that always starts with A and together starts with T. It's very highly scientific. If you can come up with a better one, be sure to let me know. That's how I remember. Sorry? Always together and graciously crooked. Can I say you're terrible at this?
Thank you.
You're welcome. So now, if I give you a strand of DNA, if we had this, strand of DNA here, and its complementary strand there. Oh, look, it's such a beautiful strand of DNA. And if we took this strand here, and I gave you the code for it, maybe I said, okay, that is A, G, C, T, T, C, G, G, A, T, A. You could write for me the complementary strand. So go ahead, write for me the complementary strand. What's the complementary strand that goes with that? Take a minute and do it. So that's super cool. Hey, keep them busy?
Oh, yeah.
Yeah?
Yeah, every day at work is a fun day. And I mean that genuinely.
Those were just, and to me, it's just nothing better. It's like, if you gotta go to work, if you're with, people you have fun with, and, you can just have a stupid fun time. Like, jobs I had at your age, I had so much
Me and my boss are a walking HR issue.
Well, I can imagine. But, hopefully not too much of a walking HR issue. But no, those are fun times. It's really wonderful.
Yeah.
Hi, guys.
No, just three shifts. I was, anyways, I was in the garage for Christmas. One of the things, right?
Yeah.
My little 3-hour burst, 8-hour, 8.5 hour shift, standard shift.
Yeah.
I was in the garage, so I show up, get suited up, get the vest on.
Yeah.
Go with my partner, we get ready, grab a shirt and go.
Yeah.
It was nice. It was cold, but it was refreshing. So I
Yeah.
ditched the jacket, right? And now I'm being identified for you, right?
Yeah.
They notice. Chasing these same-sex crackheads around. Oh, and it's getting funny because I start finding them. Now that I'm following them off property, I follow them from the 9th floor to the bottom. They're sending death threats my way, everything. Oh, so fun.
Oh, that's got to be so much of the job, I imagine. Is chasing around just.
Oh,
Yeah.
Wait till night 2.
Talk to me. What's my compliment? Yeah. Whoa. Okay, listen. I'm glad that you can move at the speed of light. I'm more earthbound here. Slow down. A. Very good. Okay, good. So if you know one strand of DNA, and this doesn't happen because the cell puts them together this way, this happens because that's the way the molecule fits. Does that make sense? If you tried to put a T here, it simply wouldn't go. It wouldn't interface with this, and it wouldn't work. Does that make sense? For the molecule to hold itself together, the guanine base is designed so that it has to be across from a cytosine base. And it won't work if it's across from another guanine, and it won't work if it's across from a thymine, and it won't work if it's across from an adenine. Now, what advantage does this structure give us? And keep in mind, of course, I'm not going to draw it. Don't draw this. But out here would be a phosphate, then a sugar, and a phosphate, then a sugar. And on this side would be a phosphate, then a sugar, all the way down. What are some of the advantages of this design? Yeah, okay. So beyond just the fact that it's coded, right? Because, yeah, keep in mind that if we call this the coding strand, in other words, if this has the actual codons that we read, this then doesn't, right? Like you could imagine if every letter in the alphabet were paired up with another alphabet, and if on like the opposite page of your book were all the other letters, just like random, that wouldn't also form another story, right? Does that make sense? So this is the coding strand. This gives us the information to read. We call this the complementary strand. And so what's the advantage to having a complementary strand? Because you're right, the coding strand, definitely. It's easy to read. We've got these 3-letter codes. They tell us what amino acid we need to build a protein so we can make machines in the cell. Cool, love it. But why bother with this complementary strand? What does that do for the DNA? I mean, the answer is maybe it does nothing. Maybe it's just a random genetic, random adaptation, but it has big advantages. Can you think of what one might be? That's right. And everyone needs a buddy, right? Sure.
I feel bad that we're ignoring your friend.
No, you're not ignoring my friend. Ivan, do you feel ignored? No. What's your language, Mr. Simpson? You can grab a seat, by the way, if you like. You can sit at my desk, you can sit here, you can grab a cup of tea. No, Ivan came to visit, which I love, and then I completely ignored him all lunch because I ended up having unexpected things to deal with. But no, I am here, and so is Ivan. Yeah, so what's an advantage here? They do, absolutely, right? Because what's on one strand determines what's on the other, absolutely. So for sure they do. But the idea, one piece is, if you look at this structure, in this molecule, where's the information? Like physically, where is it located? On the inside. Why is that an advantage? What part of the DNA molecule are we trying to protect? The information. And where is it physically located? Inside. On neither side is the information exposed. And of course, if it just lay flat like this, well, then the information would be exposed from the top or the bottom, right? But it doesn't lie flat. What does it do? It twists. And the result is that the information of DNA is always surrounded by these phosphates and sugars that act as protection. Does that make sense? And if it was single-sided, that wouldn't be true, right? It could twist, but one side would always still be exposed. So we get a few advantages. And let's write advantages of this design. And I'm not going to say design because design implies a designer, and one of the predicating ideas of evolution is that this has simply happened. This has come into being. But advantages of this structure, well, the first is protection. The information is on the inside. With the phosphate and the sugar providing protection. Okay, that is definitely a big advantage, right? If you have something as important as the blueprint of how to run the cell, you want to have that protected so that's not just going to get damaged by anything that floats by, right? And of course, our DNA is held inside of a nucleus, but earlier cells, that DNA floated free as like a plasmid in the cytoplasm. You want that DNA to be protected somehow. So this protects the important part of the DNA, the information. What else? Any like computer nerds here? Don't write this. Anyone ever set up a RAID system for their hard drives? No? No? Nobody? Just me? In a RAID system, you can have 2 hard drives, but if you put a third one in, so if this is hard drive 1 and this is hard drive 2, as long as they're the same size, you can put a third hard drive here. And then what it does is that every spot on this hard drive is the value here plus the value there. And the result of that is, if one of your two hard drives fails, you can reconstruct it from the third one. So for 3 hard drives, you get 2 hard drives and a backup of both of them. That's pretty clever, isn't it? That saves you 33% of costs if you want to have all your data stored and a backup, which if you're running like a data center, like, for Google, where you have to have, 2 million hard drives, that's a really important thing. DNA is structured the same way. So DNA gives you a built-in backup. Because if any one base pair is removed or damaged, or not base pair, but rather base, if any one of the nucleotides is lost or damaged, it can always be replaced with the correct nucleotide. Damage. So a UV ray, right? When you guys go to a tanning salon, which is the stupidest thing you could ever do in your life, or you go out and tan in the sun, which is also the stupidest thing you could ever do in your life, you're intentionally exposing yourself to ultraviolet radiation, which hits your DNA and pops off a nucleotide. Happens all the time. That's what UV radiation does to your DNA. Pops it right off. Now, luckily, if all it does is pops off one little nucleotide, well, that can be replaced. Because if we had, for example, A, G, C, A, missing spot, G, C, T, T, A, and then over here, of course, we'd have what? T, C here, G here, and then let's say there's a C here, and here there'd be what? And here? And here? Okay, what has to go here? Yeah, that's the worst looking G anyone's ever drawn. Now, will this help you if both of these get damaged? No, so stay out of the sun. But at least if only the one gets damaged, it's a built-in backup. Does that make sense? It's your RAID system. That's tremendously important. And then the third piece should be obvious here. And this is that thing we've hand-waved in grade 10, and then we hand-waved again yesterday. What happens right before mitosis? Okay? And it doesn't happen just right before mitosis. Interphase is the whole thing that comes before mitosis. But as we're getting ready for mitosis, what happens? Duplicates the DNA. What stage of interphase is that? Nope. That's where the cell gets bigger right before mitosis for the final growth before S. S standing for? Synthesis. The synthesis stage. Yeah. So the S stage. So how does that happen? Well, it happens because DNA's third structural advantage is duplication. All right? DNA's structure ideally suits it to replication, or I should say duplication, since that's the word I just used. How? Well, let's say we've got our little strand of DNA here, and I'm going to use a ... to say that, like, obviously it's much longer than this. But let's say we've got A, G, C, C, T, A, G, C, C, T, G, C, ... and then down here we would have T here. Okay, now, let's say we need to make a copy of this. Well, what we can do is basically unzip the molecule. Yeah, you want to ask first. That's important. That's very important. But, assuming enthusiastic consent, you can just sort of open this molecule up so that what you end up with is this. So now we've got like A, G, C, C, T, A, G, C, C, T, G, C. And so you need a molecule that will split this apart, right? And then down here we've got this next molecule, T, C, G, G, A, T, C, G, G, A, C, G. Okay, but now, so one molecule, and this would happen using a protein, using an enzyme, which is just a protein in the cell that does work. It actually is called a protein called DNA helicase. You don't need to know that, but its job is to break that up and open it up. And then another molecule can come along, and it can start to build the second strand. Does that make sense? But when it builds the second strand, what's it going to put here? What's it going to put here? 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 Right? And this molecule is going to make its way across and keep doing that. Does that make sense? And then we can get another copy of that same molecule, and we can put it here, and it can go across. And as it goes across, what's it going to put here? C, and here? That's a C, I think. A, and it's going to go across, right? And keep going. And what are we eventually ending up with? This one will end up being a complete strand of DNA, and this one will end up being a complete strand of DNA. So if DNA helicase, right, this little molecule here, helicase, and again, you don't actually need to know that, but if it keeps working its way down here, and then these are molecules called polymerases, DNA polymerase. Again, you don't have to know that. You'll learn that next year. But what I do want you to know is that, like, you can use one piece of DNA with pretty simple machinery. You can turn it into two pieces of DNA. Does that make sense? But the thing about the two pieces of DNA you make is that they will be perfect copies of each other. They'll be perfect copies of the first piece. Does that make sense? That's a pretty clever system, yeah.
Does not make sense.
I've been told that before. Okay, one sec.
Dot.
Oh, God. They're enzymes. They're molecules. Is it helpful if I just get rhythm? Is that?
I was confused. Did we like put something else there and then something else?
Well, the point is that there has to be a machine that does this. It doesn't just happen on its own. Does that make sense? But that's okay. What we'll do instead is we'll say this. We'll say an enzyme. Well, we'll stick with protein. We haven't used the word enzyme yet. A protein called polymerase adds new base pairs, or new bases, I should say, to each single strand. And what we end up with is two pieces of DNA, right? We end up with the original one, that's AGCC, TAGCC, and it has got its new base pairs here, which would be TCGGATC. Really wishing I'd picked a shorter strand here, but that's okay. And then what do we have at the bottom? Well, we have a second copy, right? That is TCGGATCGGACG. Now, what don't you need to know at this level? I do not need you to know that it's called DNA polymerase. I don't need you to know that it's called helicase. All that stuff's from next year. It doesn't matter. What does matter is that DNA itself is like this perfectly designed molecule. And by the way, the ... is just here showing that this keeps going, right? Because that's like a big long molecule. Like DNA isn't, what's this? 2, 4, 6, 8. Like it's not 9 base pairs long. It's, 290 million. By the way, would this take a long time for a piece of DNA that's 290 million letters long? Yeah, DNA replication takes time. It doesn't happen instantly. That's why it doesn't happen during mitosis. It happens before mitosis. I'm going to cut this into like kind of stages. So we have a strand of DNA, and that can unzip and new pairs added. And what we end up with is 2 strands of identical DNA. all of that is baked into the structure of DNA. It's built-in backup. If, I'm out in the sun, I've covered myself in, like tanning oil, because, the skin isn't going to bronze itself, people. This doesn't just happen. This, hours of weekly maintenance. It's just this look. And a little photon of UV light comes in and blasts away and says, "Ha, you're gone." Can my body repair that? Yeah, what goes there? A. Now there is some danger. What if it came in while we're still recreating here, while this is happening, and it's just about to pair up with this and this goes missing? Can it replace that? No, they're already separated. It doesn't know, right? You might end up with 2 copies of that DNA that are different from each other. What do we call that? Mutation. Yeah, does that make sense? So mistakes can and do happen. So despite these abilities, mistakes can and do happen. Yeah. Okay. Have a good day. Thank you. Mistakes in DNA replication still can happen. These are called mutations. Oh, unbearable. Why would I do that? So when bases are deleted, added, or changed, the code of the DNA changes. What are some things that can cause mutations? Well, we already mentioned one, which is UV light in general, but that as a category, all light, another way of saying light is very good. Electromagnetic radiation, that could be UV, that could be X-rays, that could be gamma rays, like Hulk. What else? That's right.
I'm not right.
You're not wrong.
Like good enough to be written down.
No, but it's related. The answer was incest, and I'm sorry for chuckling. Incest doesn't cause mutations. The danger with incest or inbreeding is that it takes mutations that are in a family line and it amplifies them. And we'll actually talk about that. It is important. But the incest itself doesn't, in other words, like the Sex Act doesn't change your DNA, right? The Sex Act takes one person's DNA and mixes it with another person's. And if pregnancy happens, then you get an offspring. So incest doesn't have to do with making mutations. It has to do with what can happen with mutations that already exist. Because we all have mutations in our body. And some of them are harmless and some of them are beneficial, but some of them are harmful. So the danger with incest isn't that it will create a mutation. It's that if you take 2 siblings who both have a mutation and now they breed, well, that makes it way more likely that that mutation gets passed on. And if that keeps happening, it becomes more a part of the population. It makes it less likely that mutation gets passed on, right? It's still like a 50-50 chance versus a 100% chance, which if you do that math over and over again, 50% followed by 50% again is now 25%, followed by 25% again, 50% again is now 12.5%, right? So naturally, if you take a harmful mutation and you put it out in the population at large, it tends to like dilute. Whereas if you take that same population, you inbreed them 100%, 100%, 100%, it grows the number of people with that trait instead of shrinking it. Does that make sense? So it's a good answer. It's just not this answer, but we're going to talk about it for sure. And it was funny because I just wasn't expecting it. Sorry. But it's a good answer. But radiation, disease. You just studied bacteria and viruses and things like that. But for example, HPV, the human papillomavirus, which infects the vaginal mucosa, can cause damage to the DNA of the uterine lining, which leads to cancers, right? It leads to cells that have damaged DNA that grow out of control. That's why you get an HPV vaccine, right? Because that can cause damage to your DNA that leads to cancers. Different chemicals. Do you know what you call a chemical that causes mutations? Yes, that's true. They are bad. They are harmful. The specific term is mutagens, all right? Or tetragens, which actually don't affect your DNA, but can actually affect the DNA of your sex cells. In other words, they can cause birth defects. Yeah. And then the biggest one, random chance, right? Just this process is imperfect. No machine works perfectly every time. And so the machines in the cell whose job is to unzip your DNA and attach new bases to it and all that jazz, sometimes they don't work properly. Sometimes they don't work and they do the job wrong. And in that case, you can get a mutation. I'm not going to give you homework today. I know you guys have your projects due today. So get those done and into me. I want to have them marked and back to you tomorrow. Whatever it looks like, just give it to me. Give it to me. Yes, I do. Yeah, of course you may.
Thank you.
Yeah, give me a second. I want to say hi to Ivan here, and then I do want to help you with your math. But you guys are good. You got time to work, so make good use of your time. Again, if you want to make sure that work periods disappear, waste your time on them. Lila Young, put that away, or I will feed it to you.